taxonomy of the Labeonines of Northeast India. Yeah, as you know me that I am like more into classical taxonomy as of now. Yeah, later we are trying to integrate and go to integrative taxonomy also. So these are some of the fishes which we get in the Northeast. So this is a general classification which we follow from Nelson 2006 or whatever. It's there. It's vertebrata, then Echinopterygiae, Cypriniformes, and this Labeonian fishes actually comes under the order Cypriniformes, family Cyprinidae, and subfamily Labeoninae. So the subfamily Labeoninae are the fishes belonging to this are small to medium-sized fishes with inferior mouths, and they are adapted to swift currents. We can say rheophilic, or the phenomenon of rheophily. Then among cyprinids, the Labeonines exhibits greatest diversity in oromandibular structure, which is the main basis, or which is one of the main diagnostic character of the Labeonines among the cyprinids. And the Weberian apparatus contacting the skull is with supraneural bones. And in Labeonines, we have the concave baxi-occipital cross-section and enlarged first anal pterygophore. Labeonines are distributed far and wide from freshwaters of tropical Asia and to even to extending to Africa. So we can find Labeonians. Then the generic level, in case of Labeonians, the generic level is more diverse than the species level rank. Means like, with exception of some of the genera like Labeo, then Bangana, Garra, but Garra now, like a new genera has been erected like Garra, then again recently Ceratogara by Kotlet. So and Serenus, these are like the genera which are like less diverse. But in Labeonines, we can see that the diversity is more in terms of the generic rank. So in the last 15 years, we can see the addition of 10 new genera here. Pseudochrysocylus, then Kian labio, Ecrocolioplax, then Hongseo, then Protolabio, then Coffeecylus, then Sinigara, Stenorincochrum, then Perakian labio, then again recently Ceratogara. So we can find like, and this addition of new genera to the Labeonines is mainly on the basis of the oromandibular structure, the snout morphology. So particularly this subfamily Labeonines, the genera keeps on changing. Then the new genera are being erected here and there. So a lot of studies needs to be done, and there are lots of ambiguities which are there in this subfamily Labeoninae. So we have to work a lot in this field. So coming to the diversity of Labeonines, especially in Northeast India, it is because of the following factors, like the tectonic setting, which is because of the collision of the Indian, Chinese, and the Burmese plates, which I think on the second day, Professor Viswanath has explained vividly. Then the Himalayan orogeny, which has led to the speciation and evolution of different groups, which inhabits mountain streams, and also the presence of different drainage systems. So these are the main factors which lead to the diversity of Labeonines. Then, however, the generic species diversity is mainly concentrated in South China region. And out of the 40 genera, approximately 40 genera, which is found all over the world, 25 genera is found in the South China region, which accounts for around like 85.2% of the genera worldwide. And 25, and along with in the regions including Myanmar and Northeast, we can find around 33 genera in this area. So South China, Myanmar, and Northeast India, there are like 33 genera. So out of the approximately 40 genera which is found all over the world. And a very interesting paper came out in 2012 where a molecular phylogenetic tree has been made on the basis of multiple nuclear and mitochondrial genes. And according to that paper, the tribe Labeonine has been again grouped into divided into four subtribes: Labeona, Garra, Osteocalina, and Semi-Labeona. But recently, Kotlet opined that the Osteocalina and Semi-Labeona these names are not available since it does not follow the ICJN rules. So this is the diversity we can see in South China area, Myanmar, and even in Northeast. And coming particularly to Northeast, in Northeast, we have got 5 genera of Labeonines. So the first is Bangana, second Syrinus, then Garra, Labeo, and Taraki Labeo. Taraki Labeo actually it comprises the fishes which were kept under Chrysocylus and Gonorhynchus. The southeast Chrysocylus and Gonorhynchus are kept under Taraki Labeo by Kotlet 2006. So the genus the Gonorhynchus and Chrysocylus are not there anymore as such. So among the 5 genera which is present in Northeast, we can see that Garra is the most diverse. And in 2014, Yang et al. Considered that Garra is not monophyletic, and we can have more generic lineage among the members of Garra if we have. So that's why I said we have to work more on that. Then the genus Garra actually is characteristic in having a modified lower lip in the form of a mantle disc. So it has got the pulvinus, which was called callus pad before, and so that it may stick to the substratum since they are rheophilic and counteract against the high water currents. And the most potential diagnostic significance of Garra lies on the snout morphology and the oromandibular structure which is present in them. So these are some of the habitats where you can get Garra. Then, according to the water current, the development of the proboscis, then the development of the oromandibular features, oromandibular structures also differs in different Garras. This is one of the habitat, and in this habitat we can find this Garra. We found this particular Garra here. Then this is how we go for field trip, very crude in Northeast India, which I have been telling in my previous talk also. There are no roads, so we have to take this kind of vehicle. Then even to the jungle, we have to make this kind of place for stay. And when we eat, we have to eat like this, because we cannot carry a lot of things along with us when we trek for a long distance and in a high altitude. So that was the thing. Then coming to the methodology for the collection, we all know we go for electrofishing equipment. You can see me here using the electrofishing equipment and nets. Then we can go for market sampling also. So these are the things. Then preservation, we do it in this method. Then holotype is always preserved in the Manipur University Museum of Fishes, but we also deposit our paratypes in JSI Kolkata, either 1 or 2. So the techniques for identifying Labeonines, these are the steps which we should follow. As I have said in my scissorid catfish, also for the classical taxonomy, the steps will be the same. So we can include X-rays as well as CT scans also, as other speakers have talked about. So these are the things we need to undergo for identifying Labeonine fishes. Then the general counts and measurements for Labeonines we follow mainly this Hobson-Dagler and Kotler 2001, but for a specific genera, we have to go for different authors. Like for Garra, we go for Niveshwar and Vishwanath 2013, and Kotlet 2020. So likewise for Bangana, Zhang and Chen, then Cyrus Roberts, then Labeo, Jeram and Das, then Tarki Labeo, Kikoto and Page. So these are the authors we follow. Then, yeah, these are some of the parameters, how we do the measurements and counts. I'll not go into this detail. Then coming to the oromandibular structure of Bangana, among the 5 genera which I've mentioned in the Labeonines of Northeast, the most modification is mostly shown in 3 genera, that is Bangana, then Garra, and Tarki Labeo. Cyrus and Labeo, they don't show much oromandibular structure modification, but these are the 3 genera which shows like highly modified oromandibular structure. So here we can see the oromandibular structure of Bangana. Rostral, in this, the rostral cap, RC, or the rostral fall is like thick, smooth, and pendulous. And we can see that the rostral cap will be separated from the upper lip by a depression at the lateral side. And this upper lip will be bearing papilla at the lateral sides, at both the lateral sides, and the upper lip will be continuous with the lower lip, which is a feature of the Bangana. Then the rostral fold or the rostral cap, they are not connected with the upper lip since there is a depression groove. So that is the feature which we find in Bangana, and there is a presence of post-labial groove here. Then coming to the oromandibular structure of Garra, which has been, the names has been revised recently by Kotlet in Kotlet 2020 in Serato Garra paper. So we see here the rostral fold. It is highly hypertrophic, and we can see that it covers the upper lip and the premaxilla of the upper jaw, and the border of the rostral fold is fimbriated or invect, and it has a lot of papilla on it. Then we can see that the lower lip as a whole is modified into the mental disc, adhesive mental disc we call it. So before, this area was called central callus pad, but in the recent paper it has been named as pulvinus, and this was called the anterolateral lobe, but this has been changed to labellum. Now it will be known as labellum. Then here, this part is the lateral posterior flap, which is changed into labrum, and this is the anteromedian fold. By some authors, it will be changed into torus. So these are the names which has been changed recently. Actually, these names were previously used by Rameshori and Vishwanath 2013. They did a lot of work on that and the nomenclature of the oromandibular structure of Garra. So Kotlet revised it and just changed some of the names. Then oromandibular structure of Tarki Labeo. We can see that in Tarki Labeo there is no adhesive mental disc, but instead there is a mental lobe present. Then we can find that the rostral cap or the rostral fold is also fimbriated, and it has got it is papillated, also a lot of papilla. Then we can see the presence of rostral flap here, and we can see that the upper jaw is connected with the lower lip by frenulum. This is the frenulum which is present. These are the oromandibular structures which are present in highly modified oromandibular structures in the 3 genera of the Labeonines found in Northeast. So these are the generic character of Bangana: blunt, pendulous, inferior mouth. Then it is separated from the upper lip Then the lower lip is separated from the lower jaw, then lower jaw highly cornified. And we get two species of Bangana in Northeast. One is Bangana Dero and Dev Devi. Then coming to the genus Syrinus, mouth is sub-inferior, rostral cap is very thick and smooth, and it is slightly fimbriated, and rostral fall without lateral lobe. And so there are also two species under Syrinus in Northeast, Syrinus cirrhosus and Syrinus reba. Then coming to the genus Garra, as we have seen, there is the mental disc present. The lower lip has been modified into mental adhesive disc. Lips are thick and fleshy, and mouth is inferior and semicircular. So, and there are like more than 30 species, I think 35, yeah, species of Garra, which are found in Northeast in the 4 drainages. Then Tarchylabio. Tarchylabio, before it used to be Crossocylus, then Gunorhynchus, it keeps on changing. So now all the Southeast Asian Crossocylus and Gunorhynchus has been placed under Tarchylabio. So the generic character is that the snout is inferior with well-developed lip, upper lip is thin and absent, it's almost absent. Then the lower lip modified into papillated labial plate, and the lower jaw with a sharp ace. And there are also two important species of Tarchylabio, Bamanicus and Latius. Then labio, as we know, the snout is inferior, thick, then the smooth rostral cap, then upper lip separated from the lower upper jaw, lower lip separated from the lower jaw by a transverse group, and then the post labial group is continuous. So there are 7 labio species in Northeast India. So I have already talked about the oromandibular structures of Bangana. Here, this was the pictorial presentation. Here, this is Bangana Dero, and this one is Bangana Dev Devi. Then Syrinus, we can see here, Reba and cirrhosis. So mouth sub-inferior rostral cap is thick and smooth, slightly fimbriated. So this is without lobe, without rostral fold, without lateral lobe. These are the features. Then this is labio. This is labio diachylus, and this is labio rohita. Then Tarchylabio. These are the mouth structures of Tarchylabio. Tarchylabio, Bamanicus, then Latius. Then this is another species of Tarchylabio. Yeah, coming to the position of the mental adhesive disc in Garra. So when we like imagine a transverse line across by joining the maxillary barbel bases, then we get these 3 things here. The first one, we call it the mental adhesive disc is anteriorly positioned. We call it that way. The second one is the mental adhesive disc medially positioned. And the third one here, this is posteriorly positioned. How do we like get this anteriorly positioned mental disc? It's like if the transverse line, what we have said, it is an imaginary line, it is not present in Garra, so we have to like make an imaginary line by joining the bases of the maxillary barbel. If it crosses half of the pulvinus is the central callus pad which authors usually use before. If it crosses half, then it is called anteriorly positioned. Then if it crosses the middle of the pulvinus, posterior half, that means posterior half, sorry, in A it is posterior half, so it will be anteriorly positioned. And if it crosses the middle of the pulvinus, then it is called medially positioned. If it crosses the anterior half of the pulvinus, anterior half, then it will be called the posteriorly positioned mental adhesive disc. So that way we can see here, these are the posteriorly positioned adhesive disc. You can see there the diagram also. then medially positioned disc in this group, then anteriorly positioned. So these are some of the Garras where you can see the difference, different mental adhesive disc, and we can group whether they are anteriorly positioned or medially positioned or posteriorly positioned. Yeah, a very interesting feature in Garra. What are these actually? What are these? Why am I showing this in my slide? Why? Because these are the stones which are kept in the shores. Why they are kept? To deflect the strong sea current or the sea or current water, strong water currents. So in that way, Garra has a very fascinating thing in them. The snout, the proboscis, we can see here the presence of proboscis. So this also acts. So likewise in the seashore, this stones acts as a vortex for deflecting and for reducing the water force and the current. Same way in Garra it happens. So when the water comes from the snout from this region, then it will go through these grooves and it will get deflected by the presence of this proboscis. So that's how they can go upstreams against a strong water current. So this is a very important feature in Garra, the snout morphology. So we will go into the snout morphology here. So in Garra, there are like so many different types of snouts present. So like I put it in this way, Garra can be grouped into 3 groups. Garra with smooth snout, Garra with smooth snout. Under this, we can have a totally smooth snout Garra, and it can be again subdivided into another second one, which is the Garra with rostral flap. And the third one will be Garra with rostral lobe. And the second type of a group of Garra will be Garra with transverse lobe group. So there will be a presence of transverse lobe, and the group can be shallow or deep. Then Garra with proboscis group. So under this, we can have a unilobe proboscis or a bilobe proboscis or trilobe proboscis. So coming to this, which I have explained earlier, when I say Garra with smooth snout, this is the feature of a Garra with smooth snout. So in this line drawing, there is, I haven't shown the presence of transverse depression, but still at the tip of the snout in this area, we can find a transverse depression in the smooth snout Garra at times. So that depression, because of the depression, it will lead to the formation of here. After the depression, there will be a little elevation with, it will give a hump-like structure. So that is there. And in this smooth surface Garra, there will be the presence of sensory pores, and we can find what do you say, this tubercles, which are minute to medium-sized tubercles. And the sublacrimal group, it's not shown here, but you can see in the next picture here, SLG. SLG is a sublacrimal group. It will be very smooth, and it will be shallow, or it can be deep in some other species, and it will be connected with the rostral cap. So here, rostral cap group, actually RCG, it should be okay. There was a like mistake there. So RCG. So that is the feature of the snout in smooth snout group of Garras. Then coming to the smooth snout group of Garras with rostral flap. What is a rostral flap actually then? Rostral flap is this. Rostral flap is actually a triangular, broad rounded feature here, a triangular shape or structure which are present in pairs. So it will be a paired structure at the tip of the snout, and it is fleshy, and it is sharp, but it doesn't have a cutting edge, and it is movable along a vertical axis. So we can move the rostral flap. Then the rostral flap bears small conical tubercles on it, and it is elevated from the surface of the snout. So the rostral flap is elevated. Then when we say Garra with smooth snout but with rostral lobe, so what is this? Rostral lobe is this here in the picture, the last diagram. This is the rostral lobe. So it is an elongate conical structure with a conspicuous conical tubercle at the tip, and it is not elevated from the surface of the snout. So this will be not elevated, and it will be encased inside the lacrimal groove. So that is the feature of Garras having rostral lobe. Then, yeah, this is how I wanted to show you, like clearly. This is Garra with rostral lobe. Actually, this is found only in one species in Northeast, Garra manipurensis. Then we have the Garra with rostral flap. Flap is here. This is a picture of Garra rachinica. So we can see what are they actually. Then coming to, yeah, I talked about this picture actually is for another group, that is transverse lobe group. Here we can see here there is a transverse lobe present here at the tip of the snout here, and if there is a transverse lobe, obviously there will be the presence of transverse groove here. So these are the different lobes present in different Garra species. Then these are the smooth surface Garra. And you can see here the formation of hump, which I said here, a little depression kind of a feature will be seen. And because of that, it is a little elevated here in the posterior region here, posterior part. And so it forms a little hump-like structure. Then we can see the minute tubercles and pores in this kind of Garras. So these are the Garras with transverse lobe. This is the diagrammatic, the art diagram of this. Is this transverse lobe, then transverse groove present. So these are elongated fleshy fold posteriorly. What is a transverse lobe? So elongated fleshy fold posteriorly demarcated transversely by a shallow or a deep transverse groove over the enterodorsal surface of the snout, just behind the tip of the snout. So this is a picture which shows that Garra arupi campi. Then coming to the snout structures, these are the group of Garras with proboscis. So the snout structures on members of Garra with proboscis. I have said Garra with smooth snout, Garra with transverse lobe, now coming with Garra with proboscis, the third group. So how does the snout of a Garra with proboscis look like? This is the thing, the proboscis is here, then there is a transverse lobe, transverse groove, then we have the lateral surface and the sides of the proboscis, then depressed rostral surface. So Garra with proboscis was subdivided in my earlier slide into unilobe proboscis, bilobe proboscis, and trilobe proboscis. Unilobe proboscis is this. So this is clavirostris, this is Garra substrictorostris, and Garra, this is substrictorostris, and quadratorostris. This is quadratorostris. Then we can see here in bilobe, we have birostris, biloborostris, and this is Garra cornigera. Then in trilobe proboscis, we can see here Garra caladinensis, then Garra trilobeta, and this is Garra paratrilobeta. So these are the different proboscis shapes and structures which are found in Garra with proboscis snout. So yeah, the shapes of proboscis can be quadrate. That's why accordingly it was called quadratorostris, then bilobed, then quadrant, then triangular here. So depends on the shape, how you called it. So you give a name to that. Then The tubercles arrangement also we can see here, how the tubercles are arranged on the proboscis and on the transverse lobe also we can see. So this kind of things needs to be observed while identifying Garra species. So the tubercles can be conical, cylindrical, pyramidal, flattened, and this tubercles can bear unicuspid, multicuspid tubercles. So this is multicuspid cylindrical tubercle, then multicuspid flattened tubercle here, then this is unicuspid conical tubercle. These are a few examples which I'm giving. So we need to look at the shapes of the tubercles and how it is born. So that is the thing. Then the evolution of the mental disc in Garra. Actually, it was discussed by Hora that it was in Garra the mental disc developed in 8 stages in Hora 1921. So we can see that in this, when we talk of the Labeonines, the different Labeonines, so after knowing what Hara said in 1921 and what we see nowadays, we can know that here, the development stage, this is the most primitive one, Serenus, then Labio, then Bangana, then Labio, Garra, and Garra. It's the most specialized ones are found in Garra. So here we can see that the first picture A, this is of Sirenus. We can relate that. So Sirenus in this, the anterior labial fold is developing and it is covering the anterior lip, and also the branchiostegal rays are extended till here. But when we see picture number, this is found in Sirenus and Labio, and the second, third, and fourth picture, they are found in Bangana and we can say Labio. So in Bangana, we can see that the disc formation, it's not there, but rudimentary discs will be formed later on, and there will be the presence of mental lobe in Labio. So here the branchiostegal ray starts expanding and the isthmus becomes broader. That's how it develops. Then later on, this fourth, fifth, and fifth, sixth, and seven, in 8 stages. These are the developmental stage which occurs in Garra. So this is how in Labeonines the lip structures are developed. This is just we presume that way. So we need to do a lot of evolutionary work here. Then, this is also a very important and interesting thing which is happening in Garra, like the proboscis, the presence of proboscis or the development of proboscis and the shifting of vent position in Garra. They are a very useful tool for studying the systematic and evolutionary process that is happening in Garra. So when I say anterior, so vent position can be measured, like vent is like if it is found a little towards the pelvic fin rather than going towards the anal fin. If it is towards the pelvic fin, then it will be called anterior. Then if it is in between the pelvic fin base and the anal fin origin, then it becomes the middle position of the vent. Then here, this is the posterior position of the vent here in this. It is towards the anal fin base. So like we can see the anteriorly positioned vent in Garras with smooth snout, and these second one, the middle, if the vent position is present in the middle, then this type of vent is found in Garras. These are correlated. It seems it is correlated, and it is found in Garras with transverse lobe. And these are found if it is posteriorly positioned, the vent, then it is found in Garras which with proboscis. So there is a thing that Hora 1930 and even Manon also opined regarding the position of the vent. It is said that if the Garra has a smooth snout, then they have to try to stick to the substratum as such. That the body, anterior part of the body is suppressed, and there is a change in the positions of the visceral organs. So because of that, the bend changed to the anterior region. It is like kind of a constricted, trying to stick to the substratum. So that is the thing which Menon opines regarding the vent position in Garras. Then coming to the surface, gross morphology based on SEM micrographs. We can see here, this is a rostral cap, then the torus, then this is the pulvinus and the labrum region. We can see papillas, which is bearing unculais. So here, yeah, if we take a closer look at this, the unculais are like finger-like here. So these are the features which we find in Garra. Then when we go for the snout of Garra, you can see here, this is a tetracuspid tubercle, which is seen under the SEM micrograph. And this is a multicuspid tubercle here. Then this actually is a protruding tubercle, which is hexacuspid. So this is non-protruding tubercle, which is inside. That's all about the identification of Labeonines, and especially I gave main preference to Garra because the diversity of Garra is very high in Northeast. That's the thing. Thank you. Any questions?